I'm going to start, and good afternoon to all of you. And more than one way to build a blackbird, the quickest of evolution in island melanism. So we choose this research article from PLOS Genetics. It's on selective strips on different pigmentation, gene-mediated convergent evolution of island melanism in two incipient bird species. And it was from the professor's campaign, Bose, Sepia, and we. Most of the professors from the Cornell University. And today we are going to present this. And next slide, please. So the convergent evolution and determinism in the evolutionary biology. So we all know the extent of evolutionary change. The change prediction is a matter of debate in evolutionary biology, since it can be differ with the evolutionary processes and other molecular targets can give different phenomena. So instance of the convergent evolution support the argument of the deterministic evolutionary change. In here, the stochastic historical events such as the earthquakes and the volcanic eruptions can be lead to the divergence outcomes. So next slide. So in here, there are some classical examples of convergent evolution we are getting, as we already know, the independent origin of wings in birds, bats, and insects. And also there are repeated radiation of ecomorphs in Caribbean lizards. They show this, as you can see in the figure. The grass bush lizards have some adaptations to adapt to that environment, and also the trunk ones, and also the trunk crown ones, and also in the loss of flight in insects and birds associated with insularities and the evolution of island melanism. So next slide, please. Since I have interested in the adaptation in island adaptations, my research is also an example on convergent evolution, and I've done about the ashated laughing thrush, the convergence of the ashated laughing thrush regarding the phylogenetic and the plumage color analysis, and from that, the genetically it was basically a babbler, but with regarding to the plumage, it was showed that ashated laughing thrush show more closely related to the laughing thrushes. So the convergence between these phylogenetic and the plumage color shows the convergence in Sri Lanka also from the ashated laughing thrush. We can see that the conversion pattern can be observed in ashated laughing thrush too. Next slide, please. Regarding this paper, so we are going to talk about the island melanism, the increase in the frequency of dark-colored individuals in isolated populations of animals on islands. So darker coloration can be seen in a variety of species like insects, reptiles, birds, and mammals. And it's an evolutionary adaptation, and it was unique to environmental conditions found in islands, basically. And there are several hypotheses benefits of the darker plumage coloration, because it can be support as an camouflage, and also for the protection from UV radiation, and also warmer condition, thermoregulation part, and also crypsis and parasite resistance. In here, in this paper, we are talking about the melanocortin 1 receptor, MC1R, and agouti signaling protein. And with this, we will find that increased deposition of eumelanin leads to black plumage of this our study organisms. Next slide. Our study organism is chestnut-bellied monarch, Monarcha castaniventris, from the Solomon Islands, and it's a complex of closely related subspecies. And it was in an early stage of speciation, and it is varying plumage color, song, and body size. And Monarcha castaniventris eugensis it's an iridescent blue-black plumage found in satellite islands, and the megalhydrangeus was found in, it's an endemic subspecies found in Makira, and the chestnut belly color was chestnut, and the upper parts were iridescent blue-black. And island melanism in chestnut-bellied monarchs, as you can see in this figure, the Makira was the main island that you can find the chestnut-bellied monarchs, as you can see in the figure here. And that was the main phenotype that you can identify in the chestnut-bellied monarchs. And within these Yuki and Three Sisters and the Santa Ana Islands, you can find that the phenotype colors varies within these islands. Although the main island is the phenotype is color basically into chestnut belly, but in the islands you can see that in Yugi and Three Sisters, Eugensis, and Santa Ana and Santa Catalina, you can find that they are black, fully black plumage. Next slide. And the work leading to this paper, the phylogenetic and the candidate gene studies, the phylogenetic analysis using the reduced representation genomic data they have used. And for the MC eugenesis individuals from the satellite islands, that UG and the Santa Ana Santa and the Santa Catalina are independently derived from the chestnut-bellied Makira populations, and the melanisms has evolved repeatedly and convergently. And also the candidate gene studies suggest different molecular bases of increased melanin deposition between the Ugi and Santa Ana and Santa Catalina populations. So in the next slide, we are going to see their aims. Their study aimed to uncover the molecular targets and evolutionary processes within these populations, and the reference genome generated for chestnut-bellied monarch and the high convergence whole genome data obtained for individuals for Makira and satellite islands were obtained for this study. And then, next slide, please. And within this research, what they have done is use data to quantify the differentiation and reconstruct the phylogenetic affinities. They had reconstruct the phylogenetic affinities between the Ugi, Makira, and the Santa Catalina, and infer demographic history of this population, and use genome-wide approach to identify variants associated with the melanic plumage, and also the infer evolutionary processes, and estimate when mutation arose, and also the time of selective events using the machine learning methods were used in this process. And next slide, please. And from these study methods, they have found that the population are independently derived from chestnut-bellied ancestors. As I told before, in the first figure A, you can find that in the mainland Makira, they had chestnut-bellied monarchs, and the other Ugi and Santa Ana and Santa Catalina, the fully plumage color was it was mediated from the phenotype is different from the mainland. And in B, you can observe this chestnut belly and how it was differentiated between three individuals, the variation between the three. And next slide, please. And here, this is the phylogenetic maximum likelihood tree they have developed for the populations. And in green color, in the top, it was the Uge population. And the red color one is the Makira population. And you can identify the blue color is the Santa Ana and Santa Catalina population. And you can identify that the recently divergent one was the Santa Ana and the Santa Catalina one, and the Makira was the mainland source population, Although you can identify that polyphyly is finding in this Ugi and Santa Catalina, even though they are Monarcha, Controvensis, oogenesis, you can identify the polyphyly in here in blue and green color. And in the figure D, the PCA was done for the, in PCA, you can identify when we done the piece, when they done the PCA, the red color one was the Makira population, and the blue one is the Santa Ana and Santa Catalina, and green color was Ugi. Although the black one is the melanic one, can be identified in both all three locations, and mostly the chestnut was found in the main island, Makira, and the intermediate and melanic are mostly found in the islands, the darker ones. Next slide. And here you can find this admixture plots. In the green color, the population Ugi, and red color also, blue color, the oogenesis, green and blue color, you can find the oogenesis subspecies. And the whole Ugi was a black phenotype is shown here, and in the blue color oogenesis, black and the intermediate one, and in the red color you can find the Makira main population. But in the genes, you can identify that in green there are some Makira gene flow has happened for the Makira genes also in the green color, and blue color also Makira, and some small genes were found in green color and blue color in the main population Makira. And this is in the F figure, you can identify the divergence between the divergence time between the populations, Ugi, Santa Catalina, and Santa Catalina and Santa Ana Santa was found in this one. And you can see that the divergence between these two populations are very high. And regarding to that, the Makira versus Santa Ana and Santa Catalina, and also the Makira versus Makira versus Ugi are very recent. They have a very little divergence. And also regarding the divergence time, according to the G figure, the Makira versus Ugi's divergence time is very high regarding to the Makira versus Santa Ana, Santa Catalina. And also the migrants per generations. You can see the Makira to the Ugi is somewhat higher than the Makira into the Santa Ana, Santa Catalina island. And there's no migration between Ugi to Makira or Santa Catalina, Santa Ana to Santa Catalina from Makira. So there are no migration between the mainland and the satellite in Ugi to satellite islands from the mainland, but there are some migration in mainlands to the satellite islands. So these are the results that they have obtained, and I invite Gihan to for the next slide, present the next slides.
Hello. Thank you, Dileka. So I will go on to the next part of the results. Hope I'm audible. Here, what they have done is they have done genome-wide association studies to identify the phenotypes, the genotypes that are that are related to the phenotypes that we see, which is the melanism in islands. So here they have what have basically so for those who are not familiar with what is this Jayasinghe, I'll give a like summary on that. So genome-wide association studies uses to compare the DNA of individuals with a particular trait, melanism in this case, to the DNA of those without, basically genome of those who without those traits, in order to identify specific genetic variations that are more commonly found in the individuals with given trait. So important sort of caveat to note here is correlation versus causation, as well as in any other association test. So a genetic variation identified by a Jiva study is only associated with the particular trait and does not necessarily cause it. So further research is needed for like sort of identify what is the causal pathways. So these authors have done that in next sort of ways. Basically, what they have done here is they have created the phenotypic variable, quantifiable variable that given a score of 1 to chestnut individuals in main population, and fully melanic individuals has given a score of 2, and intermediate individuals a score of 1.5. And Jiva's test association, so between these phenotypic variable and SZP genotypes are done by fitting a linear mixed model statistically. And they have accounted the population structure that Dileka mentioned, the 3 distinct clusters, and intra-individual relatedness as well, because these are confounding factors. And they have conducted 2 analyses for comparing Makira and SASC, Santa Ana and Santa Catalina, and Makira and Yugi. And they haven't compared Yugi and Santa Ana, Santa Catalina, because we saw in the phylogenetic tree, they are not related in a sister sense. They are polyphyletic. Here are the results for Makira versus SASC. So here we can see these are the p value, log p values, which means the higher means lower the p value. We can see highly significant peak here. So it's in chromosome 11. This is the more like zoomed out part of this peak, and you can see the MC1R. You might remember that Dileka mentioned in the introduction, gene is in this peak region. So there is a clear association there. Actually, there are like 61 SNPs, a few of which are like tightly linked to this region, and which could have a functional role because the melanin determines the melanism. Another species as well. This is the results for Makira versus Ugi. Here there are two peaks in Z chromosome, which is sex chromosome, which is also interesting because coloration actually have thought to have a role in sort of reproductive isolation as well, because to identify conspecifics, usually females like use plumage color and song and among others, but plumage color is a very important aspect, and some few other research on like this same species have found the males sort of attack the other males with the same color, not the other color. Basically, black males only attack black males, which means they don't see the other species as competition, which means they are kind of going a sexual isolation, reproductive isolation. So that is interesting, but the more interesting part is that these chromosome peak in chromosome 20, which again have the region for ASIP, agouti signaling protein. So it is also a protein in the same melanin production pathway. So here, these are the like main 2 results from the Jiva study, and that is basically agrees with the previous candidate gene studies. So this sort of the candidate gene studies, when when you uh, know a function of gene, so you basically assume that it could have some impact, and you just sequence that like specific genes and check is there a sort of a signal of selection or association. Actually, my undergraduate research was basically something like that. And the next step is to basically sequence the whole genome and see actually these genes have a sort of P that is associated with these traits when given the context of whole genome. So they sort of reconfirm the results found by previous candidate gene studies. Here again, I mentioned that although these associations are there, it doesn't mean causation. So but we can actually think there is a causal pattern because that is directly related to the function. Because we already know these genes are associated with melanism in other species like rat and others. And actually the homologs are in the work in humans as well. So the next step is to look for signatures of selection. So even though we know these are associated, we necessarily don't know when that happened and how that happened. What are the evolutionary forces behind it? The GWAS part is find in the molecular targets. The next part that we are going to discuss is finding the evolutionary forces. So what they have done here is calculated the summary statistics. The main statistic they have calculated here is Tajima's D in this axis, so in this axis, and nucleotide diversity in here. So to give you a small background, Tajima's D is a statistic that compares number of sort of intermediate frequency alleles. What would expected under natural neutral evolution if a region has undergone selective sweep, that we suspect is the case here. Favored alleles should have like spread throughout population and replace other alleles. Then the Tajima's D should be negative. So because the intermediate frequency alleles should be lower than expected in such cases. And when we talk about nucleotide diversity, it is measure of amount of genetic variation within a population, and it is calculated as the average number of nucleotide differences per site between two random chosen individuals. If a region has undergone a selective sweep, the nucleotide diversity in the region will be low, because if like positive selection are going on, the region surrounding the sort of beneficial mutation would lose diversity, because the linked the beneficial mutation and linked mutations will be fixed, meaning others would be like go extinct in a DNA sense. So so here yeah, thereby looking at both the Tajima's D and nucleotide diversity together, researchers can get more complete picture of whether a region of undergoing selection. So here the researchers have examined the whole genome data for two populations, and here we can see this MC1R gene has a like low Tajima's D. Low means negative Tajima's D, and low nucleotide diversity, which is a hallmark of selective sweep. And here, here also here in ASIP, we can see it has a like that region has a low diversity. Yes, but Tajimas D is positive, which means a different picture, which means a balance in selection. That might give of like, so it should be different. But essentially, the patterns between like these both genes should be same. But that's the interesting thing. Here, so they will address that. We will address that in later slides. And the next measure is H12 and H2 to H1 ratio, which is the same idea in respect to not just nucleotides, in respect to haplotypes. So we can see the same pattern in there as well. So yeah, but the hallmark sort of features are shown in the Santa Ana, Santa Catalina populations with MC1R mutations, but it's slightly complex in the Ugi population. Okay, so they have done the same thing with a different approach. So basically looking for evidence of selection in different approach called ARG statistics. So ARG means an ancestral recombination graph, which is a mathematical representation of evolution history of a set of DNA sequences to see how the genetic information in those sequences has been passed down from one generation to next, while also showing events of genetic recombination that have occurred over time. So the ARG consists of series of nodes and branch like here. It's a tree-like structure. It represents like a genetic lineage and a recombination event. So the most ancestral node at the root and most recent nodes at the tip here, and length of each branch in the ARG reflect the amount of time that has elapsed between recombination events. So node would be a recombination event. So it's useful for variety of applications in population genetics, and here they have used to like get an idea where like when those recombination events that leads to these selective sweeps have happened. These plots show what we call species enrichment scores. It's basically a probability of observing a certain pattern, whether it is biased toward a certain species or population. It's population in this case. So that is the species enrichment score. I will explain what the relevance see in a minute. And the RTH is the relative TMRCA half-life, which is a metric that is used to determine the amount of time that has elapsed since two DNA sequences last shared a common ancestor. So you might notice all these ideas have to do with the time since the divergence, time since the sort of selection, in other words. So what the authors have argued here is that the areas of genome that have undergone a selective sweep in a given population should show shallow genealogies, means low RTH values, and comprising of most individuals of that population, meaning high species enrichment scores. So shallow genealogies in bias towards a certain population. So here we can see that bias. This line shows the sequences. This line shows the MC1R region, here we can see these Santa Ana, Santa Catalina populations has high species enrichment score around this region, and low here at a low RTH value. Basically, they have argued that is a hallmark of selective sweep. And again here at ASIP, AS1P, it's the same picture. Basically, low frequency mutations. Basically, the AS1P also shows high species enrichment scores and low RTH scores. And that is visualized in this tree form, where we can see the branches that shows the recombination events in the genes shows a very recent and shallow genealogies. So that means the selective sweep there was a selective sweep, as we did see in the summary statistics. And actually here we can see when that happened in sort of rough sense, but it's very recent. So it's important because those associations mean nothing without the axis of time. So here what they have done is they have given this sense of temporal change into this picture. Then the next thing they have done is they actually tried to measure the strength of and the strength of selection and the time of onset of selection within these genes very precisely. So they have like developed a sort of new approach. These are like from the people, the co-authors in Cold Spring Harbor Laboratory. They are computer scientists. They have what they have done is they have simulated this like hypothetical scenario that they would expect in such a selective sweep in the same sort of populations, and they have created a synthetic data set, simulated data set, and they have used a machine learning model, which are really hot these days, to like identify patterns of selection. And since they know how that looks like, these models like train repeatedly in such simulated data sets, and they identify certain pattern that we can't see from our eyes, and they apply that model into the real data set of these two populations to actually find the patterns. And what they have found is they have found evidence for soft selective sweeps on multiple variations in these regions. And actually what is like really nice about this nice about this method is they can actually go back in time with respect to generation time and actually measure sort of exactly when the mutation has happened. So they have found these MC1R mutations are like really high, they have a really high probability of selective sweep, and which is shown in yellow bars, the yellow color points. And they have actually measured the mutations as have happened 78,000 generations ago, like 10%, which is interesting because that is older than the split between these mainland and island populations. And that means the standard genetic variations have been selected in these islands. And the same thing they have done in UD populations. But interestingly, here is that region for ASIP1P IP gene, but we cannot see any signal for selection there. But they have seen a peak somewhere near this region, but not there. So what they discuss here is these UD population has split, as we saw earlier in Dileka's like demography analysis. What Dileka showed was that UD population split was much older, and the SAC split was recent. So what they are like saying here is the selection must have happened much earlier, and the sort of the mutations are already fixed. So information about these selection events has already been lost in the genome, like a sort of a extinct species that we don't have a fossil of. I think that's the best analogy for what has happened here. So we know around this like large selection, the signal gets like when mutations get fixed, a signal would be like lost, information would be lost, but we should expect some signal around these regions because of the recombination around the regions, linkage decay. So here, what they have what they assume here is this is sort of a neutral site that is around this region, and with that assumption, they have calculated this specific mutation to be much older, and the selection that I've undergone in UD population is really old. And in summary, the study found that melanism arose independently in both islands from a common ancestor with chestnut-colored plumage, and they found that a molecular basis, which is those two genes, for the convergent phenotype, and which is different in each island. So that is interesting. And researchers found that the black plumage is likely recent and very strong selection on standard mutation around MC1R genes, and strength of this selection on MC1R is similar to that found in other species, like pocket mice and human populations in Europe, and so in Europe for not for melanin, but famous lactose genes and lactase genes, and the researchers also used these ARG based methods to date the origins of these mutations, which is older than the estimated split between these islands and the main populations. And researchers also found the evidence for selection in genomic regions containing ASIP in melanic UG population, but it's also difficult to determine exact variants under selection. There's a substitution. Actually, they have found a substitution in ASIP gene that is fixed in UG population, which could be playing a role in melanism. However, other nearby mutations, such as cis-regulatory mutations, may also contribute to this phenotype. So they don't really know. And they have estimated that phenotype to be like 174 generations before, as opposed to 78 generations in the other island. And results from like multiple population genetic approaches and suggests these selective sweeps occurred in the regions in ASIP as well, although the machine learning method didn't found that. And actually, it is really surprising. The whole thing is really surprising because the ASIP mutations are much older. So we would expect the same would happen in the new split as well, Santa Ana and Santa Catalina populations as well, because the probability of gene reuse in parallel phenotypic evolution should be very high when populations are young and closely related, because you can easily, because there are already standard genetic variation in main population, they should be like, it should very easily go to the next population, the recent splitting of the populations as well. So here it is very interesting that these they have used, like evolution has used two different mechanisms for one phenotype, and they don't really go into why, but it could be that even though it could be, we can come back to the idea that Dileka talked about in the first slides, that is the evolution actually deterministic or it's more random? So we can see even in convergent evolution, there is a sort of a random part going on. So evolution has like so many tools that can lead to a phenotype, because the selection happens on phenotype. Whatever the pathways are not that predictable when there are like different components that can be manipulated through mutations. And the reason that they think these populations use different sort of mechanisms to give rise to a same phenotype could be that maybe some mutations, because they are linked to certain other important proteins, very close important proteins, might cause to like sort of look for different approaches for the same phenotype. That's just a like guess of theirs, which could which we could actually test. What I tried to do was identify this in the same manner, the importance of SERT and DRD4 genes for adaptation to behavioral adaptation to urban environments in house swallows. So I see the sort of same ideas in my research as well, because certain certain variants are fixed in the population, so the information has lost, and how to infer that, and looking at the whole starting from a candidate gene and go to the genome and look for broader patterns and how those are related. So I think for me personally, and Dileka as well, because she has a like great interest in convergent evolution as well. For me, this also like very relevant to our ideas. I think you also got something out of it. And yeah, this is the end of our presentation, and thank you. We would be happy to answer any questions, although for some things we actually have a like great idea. We would be happy to answer by the best we can. Yeah. Thank you.
All right. Okay. Thank you. Thanks. Very nice paper. Good, really good selection. Huh? For those who are not familiar with genomics, I think maybe it's touch. I think Gihan explained it well. It's a bit genomic side, heavy on genomics, but I think it's a very good paper, good selection. I think it's clearly a landmark paper, that's for sure. Those who, if you haven't understood, if you can't, if you find it difficult to understand, you are normal. If you understood it well, then you have to be a bit worried. But anyway, for those who do bioinformatics, fourth year IIMB level, I think you should be able to understand it, most of it. And I must say that Gihan's project is somewhat aligned with this, so the latter part of the paper. So it's a good pick from the two, and thank you. Yeah, so now it's open for questions. Remember, it's a good interface between genomics, ecology, and biogeography, because the root of the genomic variation is the biogeography, and the environment kind of pushes it this and that way in different islands. So it's a neat kind of modern paper. Yeah, so the floor is open for questions and answer. I think Gihan and Dileka can answer. If not, yeah, others can chip in. Yeah, any questions? I'll have a question, since normally I ask for the first question. It's striking that MC1R normally comes as the gene in most vertebrate melanism. So I might miss the middle part. I think probably I must have missed that. So why the two islands have this convergence? Like, I can see the convergence. Yes, ACIP and MC1R kind of evolve in parallel and then giving the kind of the similar outcomes. Do you know anything about why MC1R not playing a major role in the population, in the where ASAP comes into play? Is that ASAP kind of masking the divergence of MC1R, or is it that the environment is actually playing a kind of a differential role in on MC1R in different islands? Gihan, can you help me?
That's the open-ended question they have put forward to in the paper. They really haven't answered that. They are also surprised, but yeah, it's really surprising, because when there is a certain mechanism with standard variation in a main population, because
yeah, exactly. So the standard genetic variation is the same, right, in the beginning. Yeah, but ASIP is a bit older, though. I think, if I'm correct.
Here it's a bit older, and there are like standard variation in the main population as well. As you, if you remember, in the PCA, there was a black individual in the main population as well. So for some reason, the
basal variation
convergent has happened in a different gene. They don't know really why.
I see. That's interesting. I mean, yeah, that's very relevant to Sri Lankan context as well. Right. And I'll ask another question. Otherwise, Dileka might think I'm more favoring IIMB. Dileka, a question. So this is the system where Ernst Mayr studied in about 50 years ago. Same monarch system. So do you know anything about what Ernst Mayr and the early conventional zoologist viewpoint on this monarch system in the East, with the new genomic viewpoint of it? Is it the same, or is it different? Because
Uh,
they have a full paper on that, and there they comment on the evolution of the system. Even
it's actually sir. Actually, I saw that.
Okay, that's okay. It's not a trick question. Yeah. So it's interesting that I mean it's a same. They also had the same kind of conclusion. The only thing that lacks in the previous analysis is that they didn't understand the genomics of the coloration. So the coloration, they thought it's the same. Like it's like different. Like melanism is achieved in different islands, but it's not achieved in certain other islands. But they didn't know that the roots of achieving it is completely different. Anyway, so this paper shows that the roots are different, but about 50 years ago, Ansmaya showed that it's actually, so the melanism is achieved as you move in from east to west to east. Yeah. Anyway, so it's interesting. So that's the why it is still up to explain. Yeah, to be explained. Yeah. All right. Okay. Over to others.
Can I ask something?
Yes, madam. Go ahead, madam.
Hello, am I ready?
Yes,
okay.
ma'am.
Yes. This may not.
You can see your name, Mayur Jayasinghe.
Yes, I can't show myself, unfortunately. But anyway, my question is, environmental pollutants are known to either dull or sometimes even lead to darker coloration. So my question is, would that mask any genetic underlying genetic mechanism? How would that operate?
It can occur.
Any thoughts on it? I mean, even Professor Sampath can chip in, I suppose. Anybody?
Yeah, madam. Here actually they don't really, even though like they can repeatedly see this island melanism, they here that they don't really know what is the actual pathway, the selection pressure. What is exactly? Is that the size of the predation or environment, sort of any other thing? And it's interesting because that could actually explain why different pathways, like different genetic pathways, has maybe chosen by sort of evolution. Because if some of these environment pollutions give a different selection pressure, which leads to certain genes to be selected, which is accidentally nearby the color genes, that could also happen. Because certain like let's say immune or whatever genes, and they are linked to genes that give rise to colors. So in an indirect way, that could also happen. But I haven't seen any literature on that. Yeah, just guessing. Yeah. I think, madam, that's a really interesting point of view to like maybe explain why this is happening. Thank you so much.
Madam, small
Yes.
something to add, like melanins are synthesized. So animals produce melanins within. Carotenoids, animals has to acquire, so they can't synthesize. So what you are saying, the effects of toxins or effects of environmental pollutants on ornamentation would have multiple kind of dimensions. One dimension would be that if it influenced the physiology, certain colorations or certain ornamental structures like antlers or tusks or like feather, like in animal, like birds, feather patterns might not appear because that particular process can be, synthesis of that process might be altered. So if that is the case, melanin-based colorations like this, what these guys showing, might get affected. That's one aspect. The other side is that if it is accumulated from the environment like carotenoids, one would argue that they are more susceptible for environmental pollution because they anyway has to be acquired as food. So I would imagine that more like the point that you made might be more influenced. It would be more pronounced in the carotenoids. Then the bird songs are, it's clearly showed that environment pollution actually altering the song structure. That's that's another ornament. So I think it's open-ended at this stage, but I think people started to understand it or study it. So making the
that's the thing. Yeah, I mean, people have mentioned it, but not in detail papers as such. So it's still so. Yeah. Thank you.
Tarindu is raised his hand. Tarindu, over to you.
Yeah, can you hear me?
Yeah. Loud and clear.
Okay. So I have very general question. So as you
t-shirt වගේ අඳින්නේ නේද?
ඒක තමයි මමත් හිතුවා.
මෙහි අම්බල ගාපු t-shirt මොකද තාම අඳින්නේ? අපි හිතුවා දැන් සෑහෙන මෙහි අපිට නැතිනම් සල්ලි එහෙ හොඳට සල්ලි තියෙනවා කියලා.
රෑඩ් is එකයි.
alright. කැමා ගාලද රෑට අදින්නේ කැමා ගාලද? Okay, go ahead.
Okay, so this is a very general question. So as you mentioned in this paper, a geographic setup like this, you have the mainland and several small islands as well. So in a setup like this isolation and the restrictions for migration could affect other traits as well. For example, vocalization, their body shape, the size, maybe the behaviors and the body color as well. But in this paper, they particularly talk about the body color. So my question is, why just the body color is selected for further alterations, but not other traits like vocalization, their body size, or other behaviors? Or do these researchers record any alterations in those traits as well? Or don't they discuss those stuff in this paper, or they do not observe any kind of alterations in those traits? So what's your thoughts?
Yeah, Dilek, go ahead.
Yeah. In this paper, we did.
In this paper, they have only focused on the melanism because that's what's the focus of previous studies using candidate genes. Actually, I think they are in this same system. They have done some work, behavioral and song and body size as well. I'm not sure about the body size.
I think so.
But I think they have done some work in this system, and but we didn't really refer to those citations. But I think there are some. Dileka, you saw something, right?
Actually, no. When I'm just going through the images of the chestnut-bellied and the urgencies and the megahydranthus, I saw that there's a small difference between the body size. Actually, there should be a variation, and I don't know whether they have done any examination on these the body sizes. I'm just checking on the
reference. Not in this paper, but I think they have done some time back. Yeah.
But I didn't find any vocalization also.
There should be. And interestingly, there was like behavior experiments as well for sexual selection, aggression towards like certain, that's again they basically looking at body color. I think they have used decoys or something. So they have responded aggressively to the same color individuals.
Taridu,
So
yeah, I can add that something. I can see why you are coming at, you know, the background you are coming from. Why not vocalization, right? Probably one reason is that I think these birds are less vocal than the systems that you are working on. You know, in the northern latitudes, the birds are more vocal because here they are forest birds, so they have less options for. You know, the environment is more cluttered. So maybe visual displays are more prominent. I don't know. And also this is a kind of a iconic model in the tropics that people had been studying for a while. And they use this for color. That's a model for color. I think some people worked on the little bit on the vocalization of the same model, but it's mostly for color. But I mean, Tandu, yeah, that's a, you know, that's an obvious kind of, yeah, basal question. Yeah.
Yeah.
It's a valid question.
That answer makes sense. Yeah. Thanks.
Yeah.